Another type of T cell that you're going to encounter or that your body does possess is something called a cytotoxic T cell. Or we can refer to them as TC cells. Either is acceptable. This is one of those cells that when your helper T cell becomes activated, so the cell we were just talking about on the previous slide, the general of the army becomes activated and releases its cytokines. One of the functions of those cytokines is to stimulate or to activate cytotoxic T cells. So we're going to have a look at what happens when cytotoxic T cells are activated now. Very similar to our helper T cells, to activate a cytotoxic T cell, it also needs to be activated by an antigen presenting cell. Okay, so we've got our antigen presenting cell here again, which contains a small section of the non-self antigen on an MHC class 2 marker, and it obviously has the complementary shape to the receptor on our cytotoxic T cell, and our cytotoxic T cell is shown here. If that is a correct match, then obviously we have achieved clonal selection, just like we would have if we were looking at a helper T cell. The only difference is this cytotoxic T cell cannot begin the process of clonal expansion where it replicates itself to create thousands of copies of itself until it receives the chemical signals from the T helper cell to do so. So those cytokines from the T helper cell are required to stimulate a cytotoxic T cell to begin clonal expansion where it replicates and creates thousands and thousands of copies of itself. If the cytotoxic T cell does not receive the signal or those cytokine messages from the helper T cell, it will not be able to perform clonal expansion. So sometimes your cytotoxic T cells will interact with the non-self antigen first via the antigen presenting cells, but it can't do anything else until a helper T cell with the same corresponding receptor also interacts with a antigen presenting cell with the same non-self antigen and starts to release those cytokines. So it's kind of like a two-step approach. All right, so just to quickly recap, our antigen presenting cells need to interact with our cytotoxic T cells, again with the non-self antigen on its MHC class 2 markers. If we have a correct match, we've achieved clonal selection. Clonal expansion, where our cytotoxic T cell replicates and creates thousands of copies of itself, will not happen until the cytokines from our helper T cells are released and stimulate the cytotoxic T cell to replicate. The next slide, we're going to have a look at what happens when our cytotoxic T cells do their actual job, because we haven't spoken about their actual job yet. Okay, so this is just how they got activated. Next slide is looking at what they actually do once they're activated. Okay, so our cytotoxic T cells have been activated. Once they are activated, they are going to start moving all throughout your body, and they are going to be searching for infected cells. So these are self cells, so cells that belonged to your body that an intracellular pathogen, such as a virus, has infected. And the way that your cytotoxic T cells detect this is that there is actually part of the non-self pathogen associated with the MHC class 1 marker that we find on all of our cells. Okay, so the MHC class 1 marker that we would usually use to identify our cells as belonging to us has slightly altered because this pathogen has entered it, and it now gets recognized as non-self. When this happens, the cytotoxic T cell has obviously attached or interacted with the infected cell using its receptor. Okay, so we can see its receptor here. And on our self body cell that has become infected, we have a MHC class 1 marker with part of the non-self pathogen or antigen on the surface. So that little red bit is the non-self bit. Okay, so this particular cytotoxic T cell, its receptor is the perfect complementary shape to the non-self part that is being displayed on the infected cell. If this cytotoxic T cell did not have the correct complementary shape, so it's not specific to this non-self antigen, the cytotoxic T cell would keep moving past this cell and it would not recognize that it had been infected. So it will only detect this cell as being infected if it has a complementary shaped receptor to the non-self antigen being presented on the MHC class 1 marker. When it does find that match, the cytotoxic T cell will release perforin, which will punch a hole in the side of the infected cell. The cytotoxic T cell will then release an enzyme that triggers apoptosis in the infected cell, and obviously apoptosis will destroy that cell. That process there that's just been highlighted with these bits in the arrows is very similar, actually almost identical to what we discussed with reference to natural killer cells. So just have a look at that box that I've highlighted in red on the screen here, just to make sure that you know the difference. So natural killer cells will use perforin and granzyme B to trigger apoptosis in a virally infected cell. However, they will target any virally infected cell. So they are not specific. This cytotoxic T cell will only target your cells that have been infected by the specific non-self antigen that first activated it. So if this had been a different antigen that was presented on the MHC class 1 markers that was not complementary to this cytotoxic T cell, it would not take any action against this infected cell. Whereas a natural killer cell, it doesn't care what has infected the cell. If it has detected that this cell has been infected, the natural killer cell will use this process to destroy that cell regardless of what virus has infected it. And the final thing we're going to look at is the T memory cells, or memory T cells. You might have noticed these in the diagrams that I had on the screen for your helper T cells and your cytotoxic T cells, because they're actually produced in both of these processes. So in the images at the bottom of the screen, you can see the memory T cells highlighted with the red boxes. Memory T cells are replications of helper T cells and cytotoxic T cells that would be produced during that process of clonal expansion. So when the cells are replicating themselves to make thousands of copies, some of those copies that are being made won't actually go on to perform their job. They will be stored in your body to act as a memory, so that if you're ever exposed to this same pathogen, this same non-self antigen again, your body's immune response is going to be much quicker because you already remember or have lots of copies of the helper and cytotoxic T cells that are needed to fight that pathogen or non-self antigen already littered throughout your body. So it's not going to take as long to activate them. And when they are activated, you're activating hundreds, if not thousands, all at the one time. Whereas the very first time you were exposed to a pathogen or a non-self antigen, and it was just this one lonely helper T cell or this one lonely cytotoxic T cell that was activated, it took a while. Your body is a big place. It can take, like I said, 5 to 7 days, sometimes even longer, for your antigen presenting cells to find that perfect match. And unfortunately, in some cases, your antigen presenting cells won't find a match. It's pure random chance. So by having memory cells, and lots of these memory cells stored in your body, hopefully the next time an antigen presenting cell needs to find the correct helper T cell or cytotoxic T cell to be activated, they can do it quite quickly because these memory cells have been stored all over your body, ready to act at a moment's notice.